Hi, everyone. Welcome. Thanks for joining us today. If you're new to our webinar series, welcome and thanks for joining us. Criterion Edge runs multiple informative webinars throughout the year. Today's topic is Stay Focused, the purpose of the SVR and tips for building a strong PER foundation. Our presenter today is Dr. Sarah Chavez, Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I will welcome Sarah to give an overview of our session today. Hi, Sarah.
Hi, thank you, Olivia. So just to provide a little bit of context before we get started, this topic actually arose because we've been hearing a lot of talk among other regulatory professionals about really what goes into each of these documents. You know, what are the notified bodies really expecting to see? So based on some of the recent sort of intelligence that we've been gathering from attending different conferences and being a part of these conversations, we thought this would be just really valuable information for all of us who are sort of right in the middle of these preparations. So the goal ultimately of performance evaluation is to show that your IVD performs as intended and it meets acceptance criteria. And that's how you establish that your IVD may be considered state of the art. And the PER, this is a product, but it's documenting your larger process. And while your performance evaluation process can be fairly straightforward, if you know that your analyte is very well established, how can you approach it if you don't have as much data or if you have too much data? So that's what today's webinar is all about. We want to discuss the role of data sufficiency or insufficiency, as the case may be, the importance of developing a solid strategy to establish scientific validity, and then discuss how gaps in the SVR could potentially impact the quality of your PER as a whole. And we'll end with sort of a discussion of how proper scoping can help you to maximize success when it comes to your performance evaluation process. So we hope by the end of the presentation, you'll have a better sense of how to define which information should and shouldn't be within the SVR and where that other information may appear within the larger document. And then also understand and appreciate how the systematic literature review feeds into these different documents and how it can really support your preparations by leveraging any and all published data. And then finally, we will discuss how to conduct gap assessments to assess your own readiness. And if you aren't as ready as you initially hoped, how to develop strategies to increase efficiencies in your performance evaluation process. So as we mentioned before, the purpose of the performance evaluation is to provide that qualified assessment. So before you begin writing the PER, scoping is critical. You want to clearly define the purpose of your PER. So you are looking for that qualified assessment and analysis of data to establish or verify scientific validity, analytical, and where applicable, clinical performance. But notice how they say establish or verify. You know, there are some instances where it is far more clear, the scientific validity, and some where you need to do a little bit more digging to really establish that scientific validity. So how do we do this and what do they really mean by this requirement? We need evidence. And by evidence, what IVDR is referring to is clinical data and performance evaluation results. So it needs to pertain to the device and it needs to be of a sufficient amount and quality to allow for a qualified assessment of whether that device is safe and whether it achieves the intended clinical benefits when used as intended. And that's a critical part here too. So in previous webinars, we've discussed the systematic review process in detail and described how to find that evidence. So in today's webinar, we're going to focus more on how that systematic review can be incorporated into pulling together these reports and basically linking them all together. So again, in our previous webinar, we discussed the major change from IVDD to IVDR. It's this requirement for clinical evidence. And there is actually a lot of helpful information that's packed into that Annex 7, Part A. First is the fact that they do define this as a continuous process. You are expected to develop a process to continuously collect data and evaluate that data. And so the PER is not only the product of and the result of that process, but it's also a description of how you came to those conclusions and collected that data. So at a recent conference, one of the notified bodies had a really interesting way of describing the PER, and I really liked it. So I wanted to share that with everybody. He said, ultimately, the primary goal of your first version of a PER is to lay out your case. You need to provide a very well-described, comprehensive review of how your device fits into the landscape and be persuasive that it is, in fact, state-of-the-art, it performs as intended, here are my purposes. And it should provide the reviewers with enough data to support those claims. But the rest of the time, the rest of the entire lifetime of your device, you try to prove yourself wrong. So you go back, you repeat that process, and you are on the lookout for any little piece of information that could potentially mean that one of your prior conclusions is no longer true. Or maybe there's a piece of information that emerges that sort of changes how you might think of your device and where your device fits into this landscape. But the PER itself doesn't have to anticipate any change that could happen in the future or might happen. It represents a snapshot in time. As of right now, based on my intended purpose, here is what is currently considered to be state of the art. But you should be prepared and in the process of developing these approaches that allow you to update it regularly and to track those changes over time. Because you may identify purposes that you haven't even anticipated yet that could emerge in the future. And you need to design your searches in such a way that you can capture that information in the future. So according to the IVDR, your PER has to include sufficient clinical evidence. How do you define sufficiency? That's sort of the ultimate question for all of us. The evidence is ultimately the sum of scientific validity and analytical performance and clinical performance. And so what we want to talk about today is how you can use that clinical evidence for each of these different types of documents to support that qualified assessment and how to build independent documents that are well organized and thorough, yet very focused in scope. And that will bring us to today's title. So we want everyone to stay focused in your scientific validity report. We've noticed a lot of confusion among folks within the industry and presented and discussed in detail at recent conferences about the purpose of the SVR. There's a lot of debate about how to structure this particular document within the PER. So today I wanted to frame the discussion based on these conversations that we've been having with regulatory experts at these conferences who are also struggling. So if you are still a little bit unclear, just rest assured and take comfort in the fact that you are definitely not alone. So we wanted to start with what we know from the transition from MDD to MDR, because they basically went through this exact same learning process years ago when that transition took place. The state of the art was still a fairly new concept at the time, and there were a lot of interpretations about what they were really asking for. Because if you think about state of the art, you usually define that as a device that is currently and generally accepted as good practice, sort of the best of the best. But according to the document MedDev 2.7/1 Rev 4, and we can put a link to that in the chat in a bit, the state of the art describes what is currently and generally considered to be standard of care or best practice for the medical condition or treatment for which the device is used. So what we are hearing is the concept is the same, whether it's for a medical device or an in vitro device. The state of the art can be thought of as what is generally considered to be the best practice for diagnosing a particular clinical condition or state, or screening a particular clinical condition or state. You can be state of the art without being the most technologically advanced solution on the market. But this is where that evaluation part of the PER is the most critical. We collect evidence, we define our scope, and we report, screen multiple searches for that evidence to support the state of the art as it pertains to our intended purpose. So this will include a discussion of the medical condition, alternatives, clinical guidelines and recommendations, as well as the risks and benefits associated with the use of this IVD to make a diagnosis or to screen a population. So in addition, the SVR is also expected to explain that association between the analyte and the medical condition. So these are all critical to establishing the state of the art. But I thought it might be helpful to look at a few real world examples that I thought most people are generally familiar with, even if it's outside of your current area of expertise. So let's look at a real world example first, which is pregnancy testing. So I sincerely doubt that anyone on the call here will doubt the fact that HCG is state of the art for diagnosis of pregnancy. You know, it's been 100 years. I think we're all well established there. So how do you approach the state of the art for a device that's publication history dates back 100 years? Option one is you just assume the notified body agrees with you and hasn't been living under a rock, and that your SVR is a paragraph saying HCG tests are the gold standard, end of story. Option two is you attempt to describe and document the entire 100-year process of HCG and how that technology has evolved in detail. Not anything that any of us want to sign up for. Option three, which is what we recommend, is to provide a brief overview of the evolution of the technology and the maturity of the technology as it stands right now, but stay focused. Refer back to your intended purpose, because the scope and the coverage of your SVR is built on the scope that you define in your IFU. So state your intended purpose clearly. If you say the purpose of my device is to diagnose pregnancy, that's a good start. But that's most likely not specific enough for the purpose of establishing scientific validity, because is that your only intended purpose? If you start searching the guidelines, according to the World Health Organization, there are several clinically approved uses for serum HCG testing. And those could include diagnosis of pregnancy, which is what you're claiming, but it also could include diagnosis and monitoring of pregnancy after IVF treatments, or follow-up of pregnancy and complications of pregnancy, including things like ectopic pregnancy. Or it could also be monitoring of pregnant patients with complications or suspected fetal loss. But even more recently, some of the indications that are not yet World Health Organization approved include things like diagnosis of Down syndrome in the first trimester, or diagnosis of neoplasms, including germ cell tumors, and that can even include testicular cancer in men. So as you are thinking about your intended purpose, you know, you could, if you were trying to document the entire history of HCG, you could go off on many, many tangents, and you could describe in detail how and why serum HCG testing is being used for any and all of these indications. But if that's not one of the indications you're claiming, it can be confusing, because what the notified body could then expect is that you have data pertaining to those other indications. If you don't have data on your test being used to assess testicular cancer in men, do not confuse the situation and do not mention it. So based on our understanding of the notified body expectations following multiple conversations at these conferences, the scope of the SVR is defined by the intended purposes that you're claiming. You can certainly mention the other emerging indications, but be absolutely clear what you are claiming as well as what you are not claiming as an indication. And the next tricky part is defining your alternatives versus competitors. So current methods for HCG testing are most broadly characterized as either being a qualitative test or a quantitative test. You know, a urine-based assay that's qualitative could have a typical sensitivity of 20 to 25 international units per liter. That's the analytical sensitivity, because it's telling you how many particles, how much of the HCG can be identified in a typical test under ideal circumstances. For serum, it's far more sensitive. You can actually reach closer to 10 international units. A quantitative assay is even more sensitive at 2 international units. And a typical cutoff for sort of classifying a positive test is generally around 5. So it's twice as sensitive as a urine test in that respect. So if you're looking at serum HCG testing performed in a lab, you can, in theory, achieve levels of HCG quantification as low as 1 to 2 milli international units. But an at-home test versus a lab-based test, both can be state-of-the-art, even though clearly a lab-based test is far more sensitive. But keep in mind, these are alternatives, not competitors. So again, suppose you're a urine test. How would you actually select your competitors? You want to look at the characteristics of the assay, including the platform and the analytes that are being detected. The reported rates of accuracy for a typical home urine pregnancy test range from anywhere from 33% accuracy to more than 99%. And that's a huge range. And it's due in large part to usability of those strip and cassette formats. So if you're using this as one of your performance objectives, that could certainly help you narrow your list of competitors. If you are a midstream test, you would want to compare yourself to other midstream tests as your competitors. You can mention the other platforms as being alternatives, but your argument should state that you are state of the art compared to those alternatives, but you're also establishing the acceptance criteria for your competitors so that you can essentially set the bar for your device's performance. But suppose you're actually a serum-based assay. So these urine-based assays could potentially be one of your alternatives, but your clinical sensitivity is going to be far higher. Rather than 33% potentially, you're now looking at closer to 90% and above. So if you're claiming a urine test as one of your competitor, you would be setting the bar so low that that's going to look very strange to a reviewer, and it's going to be immediately sort of a red flag, because the assay technologies themselves are so vastly different, and the levels of HCG in urine versus blood are also very different. So how does this come back to scientific validity? Because remember, we also are tasked with associating that analyte in the clinical condition or physiological state. So that's where the scary picture on the right-hand side comes in. In the case of HCG, it's actually not quite as simple as that. Even though it's been around for 100 years, there are actually 6 different variants of HCG. So an assay may be designed to detect one or more variants depending on particular clinical scenarios. If you are an intact HCG test, that's commonly used to diagnose pregnancy. But it's used in serum samples. There's actually a different isoform that's being used to diagnose pregnancy in urine samples. So in addition to having very different performances, they're using very different analytes. The analytes are not exactly the same. They're using different isoforms. So keep that in mind as you are choosing your competitors. Try to identify the competitor that has not only a similar platform as yours, but also the exact analyte that is as close to the mechanism and the technology that you are using as it can be. That's going to ensure that the notified bodies are not going to question your choice of competitors. But this is what I mean by staying focused and clearly defining that intended purpose. If you know what your IFU states, that allows you to narrow the scope of your SVR to either your urine or blood tests. But it also allows you to argue that your analyte's choice of analyte, which isoform you select, is scientifically valid based on the indications that you're claiming, and that it's also state-of-the-art relative to alternative analytes as well as alternative biomarkers that some of your competition may use. So going back to some of the basic definitions, how do we demonstrate scientific validity? So as a general principle, the manufacturer is expected to conduct these systematic literature reviews to review the available data that's relevant to the device and its intended purpose. So again, we see that intended purpose getting thrown in there. That's really what they want you to use as your point of sort of your limit. That's going to be what to always keep in the back of your mind as you're trying to develop scope and trying to anticipate what information needs to go in this document. The scientific validity can be based on one or more of the following sources. So it could be relevant information on the scientific validity of devices measuring that same analyte or marker. You can look to see how your competition is built and the specifics of your competitor assays. You can search scientific or peer-reviewed literature. You can look for expert opinions and consensus statements and guidelines. In some cases, there may be very limited, if any, clinical data. So you may need to refer to proof of concept studies in order to really establish that scientific validity. And this is especially important if you are a novel device or if you are a new biomarker. And then finally, looking at the results of some of these clinical performance studies, if you have performed those in-house. So next, we are going to talk about the searches. More importantly, how to design searches that will help you support that data sufficiency without giving you 10,000 articles to sift through. So this is a critical aspect of IVDR. They want you to show that you have conducted a robust yet unbiased search and that you've also screened the articles according to established screening criteria. And ultimately, the literature review for IVDR aims to demonstrate and document the scientific validity, and it should be demonstrated for each device that provides information on or with respect to a clinical condition or a physiological state. The literature reviews in the context of a performance evaluation are trying to establish your device within the current state of the art and demonstrate that your device lives up to the same standards as similar devices in the field. It should also seek to verify your device's continued safety and efficacy. So these are going to be ultimately the searches that are going to feed into your different documents. So next, we're going to discuss a little bit more about what these searches actually look like. So let's say you have established your intended purpose and you have clearly defined PICO terms. You know that your test, in the case of pregnancy testing again, is used only in women to diagnose pregnancy. You are not claiming anything to do with diagnosis of neoplasms or cancer. You're only looking at pregnancy. That is part of your PICO terms. Those are part of those keywords that you're going to pull in. The hope is that when you do that search, you're going to wind up with clinical evidence, which is based on a sufficient amount and quality of clinical data. But let's say you're not getting the search results you expected, or you're not getting as much data that you need to establish that sufficiency. You may actually need to revisit ultimately your search question and check the approach to your screening. So as a point of best practice, we recommend conducting 3 to 4 searches using multiple databases. And I say 3 to 4, meaning that in many cases, we actually separate the PICO search and the guideline search. The guideline search will help you identify articles which refer to professional guidance, which you can then seek out directly from those organizations' websites. The PICO search is generally a bit more broad in scope, but it will incorporate many of those terms that you have based off of your intended purpose statement. So you really do need to know that intended purpose going into the development of a state-of-the-art search. If you're trying to still decide on your intended purpose and you're trying to determine if you want to claim or if you can claim that this test could be used for other purposes, you would want to devise the search in such a way to be more broad, but that also means more articles are going to be retrieved. So the third search then is the competitor search. That's where you identify several competitors who are most similar to your device. And then the fourth search is the subject device search, where you look to see if there is any published data on your subject device. So we recommend, as you're performing those searches, incorporating mesh terms and keywords built around your PICO terms, but also the product names that you've identified. In practice, you can actually devise these searches in such a way so that they actually exclude certain studies as part of the search strings rather than you having to screen them manually. So for instance, you can exclude articles that are published in a foreign language or in a particular country of origin if that country is outside the scope. You can exclude animal studies or preclinical data. You can also limit your time range so that you're only choosing articles published within the past 5 years. But I can tell you straight away, if you're looking for something that has a very long publication history, that time range can be tricky because there may not have been a lot of changes that occur in pregnancy testing or HCG testing if you limit to only 5 years. And then finally, you can exclude by level of evidence, maybe excluding conference abstracts or letters, anything that has not necessarily been peer reviewed. So just ensure that you're researching across the correct databases and that you're devising the searches properly. Not all databases are made equally. And to find the information you need to consider where you're most likely to find that information. PubMed is an excellent place to start. It's got an extensive catalog of peer-reviewed literature and links to other relevant literature based on your search, but it's never meant to be a complete search tool. And in fact, it doesn't fully cover the European and Asian literature, and many of the articles are not open access. So if your search is very broad and you are required to screen quite a few full text articles, it can get pricey. You can have to purchase many, many articles to complete that systematic literature review. Embase contains a very good coverage of medical devices and therapies used across Europe. And the issue with Embase and Medline as well are that the searches are not quite as user-friendly as PubMed. And it can require the assistance of a medical librarian to help you really craft the correct searches. So this is something that is, if you do it well and you really think through these searches, it will save you a massive amount of time in the screening step. And then besides PubMed and Embase, it's highly recommended that you choose at least one of those additional scientific databases like Medline or Cochrane Reviews. And you can also consult a clinical trial database such as clinicaltrials.gov, the European Clinical Trial Registry, or the International Clinical Trials Registry platform. So just to illustrate what I'm talking about here, let's say you have a COVID test, which is another example I think we're all too familiar with. If you just look up COVID testing in a typical PubMed search, you're looking at close to 50,000 articles. And that's as of today. If we extrapolate this out to 5 years as searches, you're going to be looking at 100,000 articles. You can narrow the focus a bit by maybe searching for COVID PCR. That'll get you down to 893 articles as of today. Still quite a few. But then let's say you want to limit the scope to things like systematic literature reviews. If there are already papers that have been published that have synthesized this data for you and have conducted additional systematic literature reviews, it's always nice to see what conclusions they reach and then whether or not your current analysis also reaches the same conclusions in terms of the accuracy, the sensitivity, the specificity of PCR-based COVID tests. So again, I want to kind of reiterate the step where we have to screen the results of these searches because you are expected to clearly present your methodology. You need to show that you've used your PICO terms and that you've identified this subset of articles. And then as you're screening, you want to clearly define why articles are being excluded and for what purpose and at what level. Have you excluded it at the level of reading the abstract or title? Because you don't have to read every single article that is pulled down from your search. Many can be excluded as being off topic or not relevant, or maybe it's a urine assay and you're a blood test. You can make exclusions at the level of the abstract in many cases, but then there is a full text review that is required for many of these articles. And depending on how well published your particular device is, that can get really big. So the level 2 is the full text screening. So ultimately, we want you to present a very clear description of the methodology that you're using. I also wanted to point out that analytical and clinical performance studies have very different objectives and endpoints. So think about it like this. The analytical performance is focused on the analyte. That's that 20 to 25 international units. That's an analytical sensitivity. How much of the HCG can I actually pick up on? Clinical performance focuses on the patient. If I do HCG testing, how many patients can I identify as being pregnant? What is my sensitivity? But it's also dependent on timing. The sensitivity one day after missed period is going to be much different than 10 days or 20 days. So pay attention to timeframes, pay attention to particular patient populations where you may expect that the sensitivity could be altered or different. And pay attention to which analytical and clinical performance measures you want to capture. Generally speaking, the indicators of analytical performance are pretty similar or even identical across many IVDs, whereas indicators of clinical performance will vary, but they depend very strongly on that intended purpose. So if you've clearly defined your intended purpose, you have a pretty good idea of what clinical performance objectives to be on the lookout for. The guidance is provided for the analytical performance based on the CLSI documents, the Clinical and Laboratory Standards Institute. But the indicators of clinical performance vary, and they depend strongly on that intended purpose. Also note the cutoffs, because IVDR mentions cutoffs under analytical performance. So ensure that you are documenting cutoffs in your analytical performance report unless you have a justification for not. So one more example, again, that we're all generally familiar with. Think about COVID testing. If you are trying to determine your analyte and your intended purpose for a COVID test, well, first off, it depends on what you're actually looking for. Your analyte could be the nucleic acid, it could be the antigen, or it could be an antibody test. Are you looking for an antigen? If so, are you looking for which antigen? You know, different tests, they may all be antigen tests, but maybe one test looks for just the surface antigens and one looks for a different antigen as well. You know, some may use multiple antigens. That's important information because that could determine whether or not your competitor is more likely to be far more sensitive than you. If they use two antigens and you only use one, or if your antigen is prone to mutation, you want to make sure that you're documenting that. So like for instance, with the spike protein, you want to document whether or not the spike protein is the correct antigen to choose if you are an antigen-based test. You want to document whether or not the spike protein is prone to mutations. If it's the same, if you are looking across different types of COVID, are you just as accurate at diagnosing Omicron versus Kraken versus Delta using your antigen-based testing? Same thing with DNA-based testing or RNA-based testing for nucleic acids. If your gene that you have targeted, if it's prone to mutation, you have to identify how many primer sets are you proposing. And then you also need to be clear on the timing. So your sensitivity is not going to be the same in a symptomatic or an asymptomatic patient, in a patient at 1 day out versus 3 weeks out. You know, there are very specific timings with which these tests are most state of the art and most scientifically valid. So as you're collecting the data, just keep in mind, this could get very complex in terms of monitoring and tracking all stages and all indications and all populations and all of the specifics of the test technology itself. So ask yourself, what's the analyte and the intended purpose? What's an alternative versus a competitor? Are there certain potential screening criteria I could build in to help me cut down on my data? Maybe only looking at symptomatic patients versus asymptomatic patients. And then determine what are my most appropriate performance objectives and data that I need to be capturing. So now let's transition and talk about that elusive data sufficiency and how you can assess available data as part of a gap analysis. Okay, so this is a very critical aspect of IVDR. You need to show that you've conducted a robust, unbiased search and that you've screened the articles. But once you've completed the systematic literature review, it's entirely possible that you may come to the conclusion that you don't, in fact, have enough data. So what do you do? Option one is that you could go back and revisit your searches and determine if maybe your limits were too strict. Option two is to review guidance. So in many cases, recommendations will openly acknowledge a lack of data, but they could also help provide you that justification that your device is in fact state of the art, despite the lack of clinical evidence to date. For example, with COVID antibody testing, that antibody response in COVID is commonly used to assess protection levels or vaccination levels. It could be, though, how else would you do that? You know, what is there another benchmark? If antibody-based testing is the benchmark, there's going to be very limited clinical evidence sort of because there's nothing else to compare it to. It's assumed that the test is accurate when it performs as intended, but you don't have a benchmark and you don't have a standard. So in that case, you may need to rely heavily on the guidelines and the recommendations put forth by professional organizations and pull from that analytical performance in the absence of sort of up-to-date clinical performance. So let's take a step now back and look at intended purpose and how that ultimately helps you frame that qualified assessment that the IVDR is looking for and provides limits to ensure that your SVR isn't attempting to cover more than what you're claiming. So your intended purpose needs to clearly define what you're detecting or measuring. It needs to state what the function of your analyte is in terms of the actual relevance in a biological system. You should describe what specific diagnostic information, what purpose does that test serve in the diagnostic clinical algorithms. Distinguish whether or not your data is quantitative or qualitative, because that's going to directly inform your performance objectives. Pay attention to what type of specimen you are claiming, and if there are differences in the reported data, if you are looking at fresh versus frozen samples or nasopharyngeal swabs versus sputum, that's a very different sensitivity level and a very different type of specimen. And then finally, think about your intended population. So you can see here the language that you use to define your intended purpose directly informs those clinical performance objectives and the screening criteria that you're going to be using. So I put this slide together basically to illustrate that that intended purpose helps you to align your PICO terms with each of the different required searches, but it's also going to help you start to see where that data goes in each of the reports. One of the biggest challenges with scientific validity is the idea that your device data is not in that report. It's meant to be objective, and it's meant to establish the sort of benchmark by which your device will be measured. Your device's analytical performance data will go in the APR. The clinical performance data for your device will go in the CPR, but the bar by which you are measuring yourself, the standards and the acceptance criteria, that is derived from that scientific validity report and that competitive landscape. And so ultimately, each of these searches are going to feed into each of these reports, but it's important to make sure that you are clearly documenting which search the data is coming from and that you are not mixing the data. The performance evaluation will synthesize all of this information and compare your analytical performance versus the competitors, your clinical performance versus your competitors. And it helps to show that scientific validity. But each of these should serve as a standalone report. And so now, after you've finished this very important scoping step here and identifying where things go, it's time to set up your required systematic literature reviews. So this is where we're kind of leaving you and where I would refer you to one of our previous webinars. After you set up the search, you've identified which data you need based on your intended purpose. That's when you start your systematic literature reviews and you start to get a sense of what's actually out there. So in the end, that systematic literature review is going to hopefully help to provide you that clinical evidence that you need to meet the IVDR requirements. But it's just as important to document the process by which you identified that information and to ensure that that process fully supports the intended purpose and is not going beyond the scope of your intended purpose to where it can be too broad. That can take a lot of time to screen, and ultimately it wouldn't wind up in the final document. So we hope that today we've covered a lot of ground, but we hope that you have a better understanding of how to leverage that published data to meet their expectations and why a typical way of internet searching or PubMed searching is not sufficient in this respect. We also hope that you have a better understanding of how that systematic literature review feeds into the independent reports, the SVR, CPR, and APR. And then finally, the importance of scoping and getting these clear statements of intended purpose up front. That can help you with these early gap assessments to help you hopefully avoid some of the common pitfalls where you wind up with not sufficient data at the end.
Great. Thanks, Sarah. That was a wonderful, great, thorough
Yes,
overview of
a lot of ground to cover.
a lot of ground, definitely. We will now turn to our Q&A portion. So if you have questions now that Sarah's presentation is done, please send them in using the Q&A button. We have about 15 or 20 minutes to get through as many as we can. And now let's get to our first question. And our first question today is from Rupa. And they're asking, what is the main difference between CER and PER strategy crafting?
I think in our experience, because we're coming from a background of writing many CERs, hundreds of CERs. And as you're transitioning to the PER, I think the main difference I've noticed is those performance measures. In a way, they're a bit more standardized, especially those analytical performance from technology to technology and in between different organisms or pathogens even. ELISA is ELISA. So you're looking for certain types of assay or certain types of data across each. But where it differs is that, first off, with defining risk, it's not really a safety objective because in many cases, there's not a direct risk of an incorrect test result other than like a delayed diagnosis. So the way that you describe risk and benefit is quite a bit different between the CER and the PER, but ultimately the process by which you are writing these two documents and preparing the systematic literature reviews and crafting these searches is very similar.
Great, thanks, Sarah. Our next question is from Karen. Karen is asking if you have any advice on how to scope the literature search.
Yes, this is one of the most challenging parts, and if it's done well, it will save you a massive amount of time. What I would definitely recommend is working with a qualified reference librarian or even multiple reference librarians. This is a very iterative process. You want to ensure that you're being inclusive enough that you're not going to limit a potential new indication that's emerging that you want to be aware of, even though you're not describing it, you're not claiming it currently. But at the same time, you don't want to see every and all off-label use of your device and go into a massive number of articles to screen through. So we usually recommend that's why having those multiple searches set up is very helpful. A competitor search will allow you to look for your competitors being used for the intended purpose for which you're claiming. So, keeping those competitor searches very targeted. So it's the device and the indication. But then the SOTA search, the state-of-the-art search, can be a bit more broad in scope to ensure that you're not so limited that you couldn't potentially pick up an off-label use.
And Sarah, you bring up a good point about the medical librarian. If anyone here is in need of librarian services, you can definitely reach out to us. You can click that link in the chat, can give you some more information on our medical librarians and how they can help you with these searches.
Yes, we've noticed that is a game changer. Being able to craft those searches correctly and using the correct search terminology and being able to search across platforms like Medbase and Embase and Medline offer a lot more sort of targeted searches than something like a PubMed search.
Definitely. All right, moving on to our next question from Farron. How do you better pick keywords for article searches and how do you use Boolean connectors?
Yes, so this is exactly what I meant by that, the limitations of PubMed. In PubMed, you're somewhat limited in terms of how you can craft these searches because you can typically only use sort of and or connectors. You can add far more detailed connectors when you search in Medline or Embase. So we typically start with the keywords by looking at the keywords that are described in the mesh strings And so that's a good place to start. But you also need to be aware, if you're searching in Embase, that those are going to have a different sets of strings and different language. So this is the part where I definitely defer to my librarians. I typically provide them with a list of mesh terms that I've identified by looking at some very high, like sort of well-cited articles and very comprehensive review articles, some of the sort of pivotal publications in the literature. But then I defer to our librarians to help to craft those searches in such a way that we're limiting those off-topic articles so that hopefully I'm not screening 10,000 COVID articles to find the data.
All right, thanks, Sarah. Our next question is from Pernille. How do you create a literature search report for your IVD?
In this case, I would say the typical literature search report is going to consist of the strings that you used, the date that you ran the search, the platforms that you used, so the databases that you searched, and then clearly document all of the articles that were retrieved from that search. So you want to list all of the articles that you've identified for each independent search, and don't mingle them. So you have your competitor search, you have your state-of-the-art search, you have the criteria by which you're screening those searches, and you will want to document where the articles came from, which search provided each article, as well as any articles that are identified as ad hoc or sort of citation mining, any article that you weren't able to directly identify in your literature search. Be careful to document that I have articles that are ad hoc searches from guidelines, or ad hoc searches that are general review articles that I use to describe my background. You're not going to get every and all articles that you need to conduct your systematic literature review and your SVR. They won't all necessarily come from a literature search, a systematic review. It's expected that you will have some number of ad hoc articles. It's just very important that the articles that you're using to derive your acceptance criteria need to be clearly documented as coming from one of these objective systematic literature reviews, not that it doesn't give the appearance that you've carefully selected only the articles that provide data that you're happy with. So that's why documenting the source of each of the articles is really important.
So Sarah, you've talked about databases like PubMed, Medline, Embase. This next question is an interesting one. Are paid literature databases a requirement for SLRs?
Yes, in our experience, we're getting a lot of feedback that PubMed is not enough. And I think it's because of the way that it's indexed, and the lack of representation of European articles in PubMed. So as a rule of thumb, generally PubMed, Embase, and Medline are kind of the core that most organizations are using for these searches.
Great, thanks, Sarah. Our next question is from Elaine, and she's asking, should the SVR be updated during the lifetime of the device as part of post-market surveillance activities?
Yes, absolutely. So really, this SVR, it's meant to be that snapshot in time, but it's also the process of writing this SVR. If you find that, there's a particular an interference that's been identified, or a reason why your test might not perform as well in a certain population, maybe there's a cross-reactivity with a certain medication, or certain patients are going to be at a higher risk of a false positive or a false negative result. Your post-market surveillance activity should pick up on that. And if you start to notice that needs to be updated within the SVR. So the first draft of the SVR is always the most time spent because you are building it from scratch. But you do need to be prepared. This is going to need to be updated, and you need to document that you have updated it every time you are submitting an update.
All right, thanks, Sarah. Our next question is from Hussein, and he's asking, for a new IVD development, how can the SVR be built to not only comply to IVDR, but also be of added value for the project decisions?
Yes, so this is something that Husam and I have been talking about quite a bit with a lot of these projects. And this is something I hadn't really considered, but this SVR is so helpful to the R&D teams. It's incredibly helpful because this is that intelligence that you're looking for. If you are in the process of designing your product, it's tricky though, because you are building all these searches off of your intended purpose. But if you're not yet settled on your intended purpose, you have to start somewhere. So you kind of devise an intended purpose, you devise these searches that are a little bit more broad in scope than you would if the intended purpose was well established. So the searches will be a little bit more broad, but it will also help you identify those competitors and how they are being designed. So if you're starting to notice, hey, most of my competitors have added a third set of primers because there's a concern that the new variant of COVID has mutated and that there's a risk of false negative results. So by adding this third primer set, I increase my sensitivity by a certain percentage. If you notice that there's a certain, a way that the field as a whole is moving, it can allow you to make those decisions in product development. So what we're finding with multiple clients and companies that we interact with, they actually do this SVR first, even often before the device is fully completed, like early in that development process, because it gives you that intelligence that you need to make sure that you're not designing something that's already not state of the art. It gives you kind of a preview as to whether or not your device will be state of the art if you can meet sort of who your closest competitors might be and whether or not you are state of the art as you stand right now, or if you need to tweak your design before you get too far into the process.
All right, thanks, Sarah. Great example. This next question from Joy is an interesting one. Why must we separate the SVR, CPR, and APR, and why not include all three as parts of the performance evaluation?
So yes, so they actually will all be sections within the PER, but the idea is that these can be standalone reports and then they will be incorporated within the full document. The easiest way to think about it is like if you're writing your thesis, you publish articles during the course of your research. You publish independent articles. They each have an independent thesis and, your purpose, your hypothesis, and your results. But if you were writing your thesis, you wouldn't just staple your 3 papers together and call it done. You have to synthesize that information and connect it. So the PER is sort of that thesis. You're describing, I did this experiment and I came up with this data for this purpose. I did my SVR. That document describes why my analyte is associated, all the data that I have of how it's associated with that clinical condition, and it sets my competitive landscape and the acceptance criteria. My clinical performance report has all of my device's clinical data. Did I meet what my competitors claim? And if I didn't, why didn't I meet it? Maybe I'm slightly less sensitive, but I'm a at-home test, and I happen to be the only point of care test available. Everything else is laboratory based. So yes, I shouldn't be as sensitive, but it's okay because I offer this additional benefit. So the PER describes things like your benefit and the risks and the benefits and that association between these documents. But each independent document should stand alone, but then be sort of connected within that PER.
All right, we have a couple minutes left, so if you haven't sent in your question, you should do so now. We'll try to get to it. This next question is from Donald, and he's asking, how does the SVR change if it is an accessory to an IVD like an antibody?
So in this case, the approach is generally the same, whether you are looking at the whole kit, the whole test, like the ELISA, or one particular antibody that is used in that ELISA. You still want to describe sort of how that antibody is associated and how it's being used in clinical testing. So the main thing, I guess, is sort of the overall focus. There's less focus on the technologies and the platforms in which that antibody is used, but more comparison between alternative antibodies or alternative methodologies that could be used if that antibody wasn't available. Like, how would you make a diagnosis if your antibody wasn't available?
All right, thanks, Sarah. This next question is a good one. This is from Beth. For IVD products not currently required to have reports, what are some simple ways to start preparing for submission in 3 to 5 years?
I would say starting with that SVR, and this can be really tricky, especially if you have like what is in the Q&A box right now. Several people have asked about devices that are in development or devices that are more generic, like a reagent kit. Getting a good sense of how the field is evolving and the maturity of the technology early in development is very, very helpful. So I would say start with the SVR, do those state-of-the-art searches and the guideline searches. Then start to get a sense of that competitive landscape. Start to see what your competitors are using. Have they slightly changed their technologies or their platforms? Are there directions or trends that you're observing? And then start to take a peek of whether or not you have any clinical data on your device, because in many cases, especially for Class A, you're not going to have clinical data, and it may not be necessary. But we're noticing that even for devices that you would think would be like a legacy that have been around for a while, there's not a whole lot of data out there, and that can be really challenging. So identifying gaps in your own product's data, and whether or not you need to plan for a trial but then you can plan your trial based on the information you're collecting from those competitor trials and the competitor data that's been published.
All right. These next two questions were pretty similar, Sarah, so I'm going to put them up together. The first one is from Dorte. Can the principles for writing the SVR be used also for medical imaging software device? And then Samantha is particularly interested in how the approach for software as part of an IVD would differ than for our hardware or reagent. So Sarah, could you speak a little bit about this topic?
Yes, yes. So when you're writing the SVR for a medical imaging software, or really any software as a medical device, a lot of the principles are similar. You still need to establish where that fits into the larger landscape for diagnostic tools, but the performance objectives may be very, very different. And that can be challenging, is knowing what types of performance objectives are commonly measured and reported for your competitors. So while I would say the approach is the same, there are going to be some nuances as to how this actually sort of gets presented within the SVR and also within the other supporting documents. So our company president, Lori, and one of my colleagues, Stacy, are actually presenting on this next week. So if you are interested in software as a medical device, I would highly encourage you to listen into their webinar, because they're going to go into more details about sort of the feedback that we're hearing from notified bodies and perhaps some of those nuances.
We are out of time. Thanks again, Sarah, for this great presentation, and thanks everyone for joining us. And we hope to see you next time. Have a great rest of your day.